Hello, Robert Barnes, Wishbone812 in here, and welcome to a video I made for you. It's an attempt to help you with your lawsuit for the Children's Health Defense. What I would like to go over is a little bit of cancer biology to maybe bolster your case just a little bit. I should be able to prove from a theoretical standpoint that the COVID shots 100% cause cancer. It's actually not too simple. But before we get into that, I have to first congratulate you on being the first person I have ever heard, beside myself, to mention this.
Viral family with no history of vaccine success and attempts to attack a virus that continues to mutate in ways prior vaccine studies don't even address.
That's always been my biggest beef with the vaccines or, the gene therapy drugs is that you can't vaccinate for single-stranded RNA viruses because they mutate too fast, which causes them to bypass vaccine-mediated immunity. Here's actually an interesting example of someone who had immune suppression because they have AIDS. And they had coronavirus for like 6 months, and they actually ended up getting multiple strains of it because it mutated so much inside of them. And there's actually several studies where that has happened because this thing just mutates super fast. AIDS actually is the same way. Most HIV patients end up having like 5, 6, 7 different strains, which is why there's never been an attempt to make a HIV vaccine until, you guessed it, after we did the whole, nonsense with the coronavirus ones. Now you're working on a mRNA gene therapy poison, supposedly for AIDS, but it can never work on AIDS. AIDS reproduces and mutates even faster than coronavirus does, so that's literal insanity. Pure poison, it'll never work, it can't work, there's no reason for it to ever work. Just want to reiterate that is, in my opinion, one of the strongest arguments against this. It just can't work for any reason, which is why it causes so much problems with the immunity. Now, a really cool thing about B cells is they actually mutate themselves. So, once a B cell, most of them just sit around. You've got like 100 billion of them, literally 100 billion at any time. They'll sit around waiting to bind to something. If they don't bind to something, they just eventually die, and then your bone marrow would make new ones. And they all have like a random sequence. There's this really cool process to go through to make a random sequence, so they just have random antibodies. But anyway, if they ever bind one, they start proliferating, so they'll start reproducing really fast. And then they'll hypermutate themselves, so they'll start like blasting their own DNA and mutating themselves, and then fixing themselves. And then the ones that bind better will reproduce more, and the ones that don't bind as well will stop reproducing. So basically, your B cells, their antibodies get better and better at binding things. Now, that's great for producing natural immunity, but if someone has the COVID shots and they keep producing alpha spike proteins, well, any B cells that they make, like so let's say they get the Delta or the Omicron variant, they start making Delta or Omicron antibodies, but those B cells keep attempting to mutate to bind to the alpha spike proteins, which are still in their blood because it got put into their DNA by the gene therapy. That's why you're getting. So people that have had the gene therapies, they can never get natural immunity to Delta or Omicron because their B cells are constantly hypermutated to rebind back to the alpha spike protein, which the alpha strain is too different to the modern strains, and so those antibodies don't work anymore, which is why you have such an increase in Omicron reinfections. People can get Omicron multiple times if they've been vaccinated gene therapied, if they've taken the Fauci juice, and they basically they can never get natural immunity to these coronaviruses. And so with that, we're going to move on to our crash course in cancer biology, which I took back in college, which was, a long time ago. So we got this fancy graph. We're not going to use that. We're going to use one I wrote instead because there we go. That's much more manageable. And, we're going to do the short version here. So basically, you got a cell division protein. I'm not even going to tell you what its name because you don't care. Nobody cares. But basically, when you get lots and lots of it, the cell will suddenly switch gears and it starts replicating DNA, and it goes. Once that kind of switch flips, you can't switch it back. It'll replicate the DNA and it will divide after. Basically, one thing to look at first is I have a little circle there, a negative feedback loop. Cool thing about tons and tons of proteins in the body, which is another reason why macroevolution is retarded, and it just shows that this is an engineered system. It was created by a designer. Is that this protein, like many others, it will actually turn itself off. So the more of it you have, the more it stops transcription and transcription of this protein. So basically, it's a negative feedback loop. So the concentration is always going back down. So basically, in order to get it to get to the replication phase, you have to have lots of inputs that make it go up. So some of those inputs are high ATP. That's ATP is a high energy molecule you get from burning sugar. Inflammation is it's caused by cell damage from immune response, things like that. Basically, inflammation is your driver of cell division. That's why if you cut yourself, it gets red and angry and inflamed, and then all the cells reproduce and it heals itself. So that's one of the primary drivers of cell division. It's also why you should not ice injuries, because ice actually reduces the inflammation and prolongs the healing response. And that's just another thing in the whole medical universe where doctors are idiots, because it's like they didn't even bother to listen to the biology that they studied before they became a doctor. But okay. And things like insulin, which is, the hormone you secrete when you have too much sugar in your blood. Actually, it causes those cells to take in the sugar. But what it also does is it upregulates their cell division proteins so that if insulin binds to them, they'll start dividing. So it uses that energy in a way that is effective. Anyway, things like DNA damage or having low ATP, where ATP is that energy molecule, those will actually downregulate this cell cycle protein so that it does not reproduce. Because obviously, if it gets a lot of DNA damage, it doesn't want to reproduce. It wants to fix that first. So this process, for it to switch from a normal cell into a cancerous cell, you need to have a few knockouts in this cell signaling pathway. Either if the negative feedback loop is knocked out, or if DNA damage no longer causes downregulation, or if it pretends that there's inflammation all the time. It's stuff like this. Basically, you have to have, in general, you have 2 or 3 knockouts to the cell signaling pathways for this that cause the system to be turned on all the time. And those knockouts would have to be caused by mutations for the most part. Now, if that's the case, then this cell can just start replicating forever. Also, you need to have telomerase turned on permanently as well, which that's complicated, but it's another mutation you have to have, or the cancer cells will kill themselves eventually. Because telomerase is necessary for cancer cells, because it replaces the caps of the chromosomes that get cut off every time you replicate. So if you don't have telomerase turned on, then these cells have a limited number of times they can divide before they, just run out of genetic material. All right, so anyway, and those telomerase generally gets turned on through a variety of different ways, but mutation in the proteins that kind of turn on and off genes, that's one of the major ways for that to happen. All right, so next we're going to go over something called apoptosis. This is basically like a programmed cell death where if the cell figures that something is wrong, it will kind of eat itself and release its insides into the environment in a way that is not inflammatory. It's not going to drive all the other cells nuts. It just kind of kills itself quietly and just lets its nutrients out. So for this time, we have to actually go over every single thing in this picture. Just kidding, we're not. No, no, we're not going to do that. Okay, here's my picture. My picture's better. All right. So again, for the arrow at the bottom, if it reaches a certain amount of this suicide protein, if the concentration gets to a certain level, it starts a process which is you can't reverse it. It just kills itself. It eats itself alive and kills itself, and sends its nutrients out into in vesicles. Very very clean death. But okay, so very similar to the other one, it does have a negative feedback loop. So the more of it you have, the more it will turn off its own transcription. That way, as you basically make little amounts of the suicide protein, as various things happen in a cell, you have to really kind of poke it a lot in order for it to kill itself. Things like inflammation and insulin, which upregulate cell cycle, like causing the cell to actually divide. Those downregulate the suicide protein, so that you can have things like healing without it killing itself. Now, at the same time, just the simple act of cell division actually generates a lot of the suicide protein, and that makes this an anti-tumor protein. So basically, if a cell just starts dividing rapidly, like if we take a cell from before where its cell cycle is disrupted and now it just keeps rapidly dividing, those cells will eventually kill themselves. They'll get too much of the suicide protein and they'll just commit apoptosis. They'll be like, okay, I'm messed up, so I'm going to kill myself now. Which is really cool that your cells have these proteins. Otherwise, you would be laden down with all kinds of benign tumors all the time. But you're not. So this is good. So another example of things like cell division that increase the suicide protein is actually your DNA repair enzymes, which I could just talk forever about DNA repair. It is one of the coolest functions of the cell and really shows that the cell was designed by an extremely intelligent creator. It's super cool how it can figure out that its DNA is damaged. It can figure out how it's damaged and repair it in whatever is the most logical fashion to attempt to retain the original information. Yeah, it's just genius level stuff. Anyway, but each of those DNA repair enzymes, if they're used and as you make them, which they're usually made by a detection mechanism where the protein will crawl around the DNA and detect looking for errors. If it finds errors, it'll start making DNA repair enzymes. And then those enzymes, they actually increase the suicide protein. So what you'll see, this is actually what a sunburn is caused by, where the sun radiation caused this really bad type of DNA repair, where you need to actually cut the DNA and then replace it and then kind of sew it back together. And if that happens a lot inside of a cell, the cell will just give up. It just gets so much of the suicide apoptosis protein, it'll say, you know what, my DNA has been absolutely hammered, I'm just going to kill myself instead, and that's what happens. So a sunburn is where that happens on a massive scale. And then other things that can cause it to kill itself is if the oxygen gets too low, it'll have a super spike in this suicide protein. Same thing with if the cell, for some reason, can no longer touch other cells. If it's no longer touching other cells, it'll be like, bro, where'd you go? I must have gotten accidentally cut off from the body. It'll kill itself. And another thing really important and interesting is T cells, which is a type of immune cell. If it is crawling around your cells and the T cells like, hey, I think you're cancer, or I think you have a virus because I'm feeling you and you feel like you've got foreign proteins on you, it will release these little signaling molecules called cytokines. And the cytokines will make a massive spike in the suicide protein and the cell will kill itself. So that's how your T cells actually defend you against viruses and against cancer, which is why HIV positive people can get weird types of cancer that most people don't get, because their T cells get killed by the AIDS. But yeah, and also that's how, just on another tangent, but that's how necrotizing fasciitis, which is a flesh-eating Streptococcus pyogenes, basically a bacteria that causes it. What it does is it has this antigen that tricks your T cells, causes them to all go nuts, and then they just run around shooting these cytokines at everything, and that's where you get the flesh-eating bacteria, where basically it just causes your T cells to just kill everything. So that's like the worst kind of disease you can get, a flesh-eating strep. So that's how that works. But moving on. Obviously, to get cancer, this pathway cannot be functioning properly. Otherwise, it will detect that it's reproducing too much and it will kill itself. So you got to have a few knockouts. Either the negative feedback loop has to be knocked out, or several other knockouts have to occur somewhere in this cell signaling pathway. Usually, again, you need like 2 or 3 knockouts. Where a knockout involves damaging one of the genes for one of the proteins in this cell signaling pathway, where that signal no longer functions due to a mutation. And lastly, in order for you to get a tumor to actually get big enough to be a problem, which you would need in either a normal tumor, just a benign one, or a malignant one, the cancer ones, they both have to have something called vascularization, which is caused by if there is a mutation in those cells where they have this angiogenic factor that they kind of release out into the interstitial or outside the cell. It will cause blood vessels to form around those cells, and you need that in order for a tumor to grow. If there's no blood vessels invading into it, then it won't get all the oxygen and sugar that it needs to constantly reproduce really fast. So for the development of your most basic cancers, you do need uncontrolled cell cycle, you need the apoptosis to not work, you need vascularization, and telomerase turned on. So you need like 2 to 3 mutations for the cell cycle, 2 to 3 more for apoptosis, at least 1 for vascularization, and at least 1 for telomerase. This is just to get a benign tumor. The malignant ones are even more than that. You need more mutations for them to like start crawling around like they're a white blood cell, for them to differentiate. It's all kinds of nonsense to get like the really bad cancer cells. But you need at least those like 6 to 8 mutations just for a benign tumor. And that finally gives us all the base information, the foundation we need in order to understand why the mRNA vaccines cause cancer. The first is recognizing that anything which mutates the DNA basically also can cause cancer. Because anything that randomly mutates the DNA has a chance of randomly damaging any of those cell signaling molecules or their pathways. Now, this information, along with the study that shows that in humans, those cells are in fact using a reverse transcriptase to overwrite their own DNA with the mRNA from the shot, basically shows that this is causing random DNA damage. Not only just random DNA damage, but random DNA damage that causes the cell to produce spike proteins. Yeah, it's really horrible for you. But because of the nature of the reverse transcription, this is, in fact, causing random mutations, so it's massively increasing chances of cancer. However, that's only half the problem, and half the reason for why this is cancer-causing. You see, with the cells that are transformed into alpha spike protein-generating cells, these cells actually are producing spike proteins and then releasing them, which spike protein causes massive localized inflammation. And what does inflammation do? It upregulates cell cycle, causing cells to divide. It downregulates apoptosis, causes the cell to not kill itself. And it causes increased vascularization. There you go. That is 1, 2, and 3 out of the 4 necessities for cancer, which is why there is a massive correlation with chronic localized inflammation and cancer, which is exactly what you will expect from a person who is subjected to a gene therapy that transforms normal cells into little factories for alpha spike proteins, which is a inflammatory cytotoxin. So to recap, the Fauci juice causes massive inflammation in localized areas for long periods of time, which definitely increases your chances of getting tumors and cancer. And also it causes mutations, which, again, increases your chances of getting cancer. Which, by the way, the word oncogene, like people say, oh, do you like have any oncogenes in your, your family history or whatever? That's just the term for a knocked out cell cycle molecule. So basically what we're talking about, how you need to have like 2 or 3 different cell cycle gene knockouts in order for you to, say, have an uncontrolled cell cycle or have no apoptosis function. That's what an oncogene is. It's one of those knockouts. One knockout isn't going to do anything by itself, but if you get a few of them in one particular cell, you can start getting some really bad stuff. So that's enough for this video. And Robert, I just wanted to volunteer my free time. If there's anything I can do to help the lawsuit, do any research or whatever, you just let me know. You can just message me on my Twitter and just let me know. And last thing is you should watch this video because it's great, and then tell people about it. Just don't post it to Twitter so that you don't get banned. All right. Have a good night. See ya.